Good evening, everyone. We'll just take 2 minutes to start. I just let the attendees come and settle in, and we'll start exactly at 8:02 p.m. Yeah, I see that more people are coming in. So once it stabilizes, we can start, I guess. All right. I think time to start. I think others can catch up. So, welcome all to the third episode of the LGP ThoughtLab series. So, I hope you have seen a previous series. If you have not, my colleague Manisha will put in the link to the videos of the previous one so you can go back. It's quite interesting. But LGP is Lodha Genius Program, and it's a program intended for high schoolers from 9 to 12 standard, and which is going to happen in Ashoka University, which is a joint initiative of Ashoka University and Lodha Group. So it's a program intended for students to come into science and mathematics, get a different flavor of it, get a sense of the real world, mathematics and science. Starts with the summer, goes into continued learning, and it's a year-round program. So if you want to know more about it, there is also a website you can go and visit. But the Thought Lab series is a special initiative, which is an offshoot from LGP, which is intended to be a conversation between people from different walks of life or science to throw some light on certain aspects which you don't get to read on a website or in a paper. So the idea is to also engage with all of you guys as audience, which is very mixed, I guess, to basically take you through the different perspectives of sciences. It can be anything. And also to enable you to interact with people who are experts in this field. So it's a two-way street. It's not a monologue. So we have wonderful speakers tonight. I think one of them, all of you, if you have seen the video or otherwise, would know very well, is Professor K. VijayRaghavan, who's also the chair of the Science Advisory Council of Ashoka University, who would be leading the conversation. Over to you, Vijay.
Thank you very, very much, Ambika, and a warm welcome to you, Marty. Professor Martin Chalfie is with us today, and he is a professor at Columbia University. He's a renowned American scientist known for his contributions in neurobiology and the development of the green fluorescent protein as a biological marker. More about that in a second, but I will briefly tell you a bit about, why such structures, such adventures in science are important. And there's no better way to say that than to quote Charles Darwin. And what Darwin said was that there is this grandeur in this view of life with its several powers, having been originally breathed into few forms or into one, and that whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Now, this is something extraordinary which Darwin said, and it tells us, as you can see this beauty on the screen, it tells us what he saw by looking at nature all around. But he, at that time, could not see in such similar amazing detail the insides of organisms. And over the last 100 years, increasingly, we've been able to do that better and better and better. And one of the extraordinary ways of doing that well is through the use of the green fluorescent protein, which Martin Chalfie and his colleagues with whom he shared the Nobel Prize brought that into general use. Marty pursued mathematics initially at Harvard University and switched to biochemistry. And then he graduated in 1969 with a bachelor's degree and returned after a period of working in various jobs, returned to Harvard to do his Ph.D. With Robert Perlman. His postdoctoral work was in the Laboratory of Molecular Biology in Cambridge. And that was an amazing place. It was at that time filled with top quality American postdocs whose fellowships from US fellowship organizations were typically more than what the British scientists got as their salary. But that was an amazing period, and that work developing the worm Caenorhabditis elegans as a model system led by Sidney Brenner resulted in extraordinary discoveries and many, many Nobel Prizes. Sidney Brenner, Bob Horowitz, John Sulston, and Marty Chalfie. And of course, that has continued to add extraordinary learning in multiple ways, how cells live and die. And so it's been really an example of what Darwin said about how life breathed into one form early on in evolution, evolves and tells us about all forms going ahead. Marty is now a professor at Columbia University, where he joined in 1982, and his research centers on neuronal cell fate determination and mechanosensation. And over now, let's get started and learn from Marty about what he's been doing since his undergraduate life in terms of research and how one can learn about what science is and how it's done. So Marty, welcome again. So here's my first question, and we'll go right to it in the interest of time. What was your first research experience as an undergraduate, and what would you tell students who face early failures in their scientific endeavors? And remember, our audience is composed significantly with school children and others who may not be familiar with biology.
Fine. Vijay, thank you for the opportunity to talk. This is going to be a lot of fun. I'm looking forward to your questions and the other questions that I'm sure will come up as well. So the first thing anyone should know is that I am not a role model. Don't do what I did. As an undergraduate, I was initially, as you said, interested in math, and then suddenly realized that I didn't think I was that good to be able to do math. And so I was looking for something else. And I got interested in biochemistry. I think, among other things, it was the hot new area. It was, it wasn't this old chemistry or old biology or old physics. It was biochemistry. So that sounded intriguing. And I knew that if I was going to continue, I should go and work in a laboratory. And I asked around, and I was fortunate enough to get a position. And that's when the mistakes really started piling up. Because I had applied somewhat late, I was placed in a room that had been, during the year, a student laboratory. There was no one else there, and it was about 50 meters away from the main lab. So I was all alone. This was not so much of a problem, but it was coupled to the other, one other problem, and that was I had this lunatic, ridiculous idea that true scientists always knew what they were doing. They were able to think out their experiments. They did it alone, and that was it. And so I felt if I asked anyone for help, that was a clear sign of failure and my inability. So I didn't ask anyone how to do the experiments. I might talk a little bit with other people to get some other ideas. And it will surprise no one that absolutely nothing I did worked for the entire summer. All experiments failed. Even the ones that didn't fail, I thought failed. So I didn't even know what a good result or a bad result was. I just was completely ignorant about things. And at the end of the summer, the person in charge of the lab said, you know, you've worked hard. There's a couple of weeks before school starts again. Why don't you go home and rest and have a good time and come back rested and try the experiment one more time? Because if you do it one more time, if it doesn't work, we're going to have to stop the project, but you should try it one more time. Oh, I went home. I had a great time with friends and family, came back well rested, and did the experiment one more time. And it failed completely. Nothing worked. And I took this to mean I should not be a scientist. Whatever my interest was, I had proven to myself that I was a complete and utter failure. And so in my last year of college, I took all sorts of interesting courses that I really enjoyed, but I had decided I was never going to be able to go into science. And uh, so my way of dealing with failure was to quit. I didn't persevere. I didn't try to, overcome adversity. I just quit, did something else. And I was very fortunate. One of the things I did was teach high school. And I made a discovery when I taught high school. And that was that the students had the summer off between years of high school, but the teachers had to find a 3-month job. And so I had to find a job. And a friend of mine pointed out somebody who had a laboratory and suggested that maybe I would go and work sort of as a technician for a couple of months. And I went to work there. I figured it was only going to be for a couple of months. I was not going to be a scientist, but I could at least maybe work in a lab. And a couple of things happened. The first was when I interviewed for the job, the person's telling me about their work, and it reminded me about a paper I had written for a course when I was an undergraduate. And I said, I remembered the result of the paper. And I asked, was that also happening in their case? And the guy that I was talking to was so surprised that I asked a question. And he said, you're the second person in 2 days to ask me this question. I think because I asked him a question, he hired me. So this was spectacular. And then he did something very interesting. It was 1972. There's no email. There's no faxes. There's just regular mail, but he didn't want to get any regular mail. He said, see ya. I'm going to do my own research in Paris. Goodbye. And he was gone. What made this time different from the first time was that I started asking people, how do you set up the experiment? And I found that other person who had asked the same question that I had. And I said, how would you test that? And so we talked about that. And by learning from other people and asking questions, I did something very risky. I did the experiment I thought of. I didn't do the experiment that the head of the lab had assigned me. And to my great joy, my experiment worked. And I did a bunch of other experiments. And the one thing I could do was go to the library and find papers on the topic. And so I did research in the library. I did work in the lab. At the end of the summer, the guy came back and asked me what the results were from the experiment he had expected me to do. And I said, I have no idea. I didn't do it. He was a little surprised. And I said, I said, but I did do these experiments and I found out this information. And I showed him the various results I had. And that became my first scientific paper. So as I say, don't do what I do, but that's what happened to me. Being able to come up with an idea, even though I could go into how horrible the idea actually was and how mistaken I was, but just the idea of thinking that I might be able to come up with ideas, that by talking with people and interacting with people, I might be able to actually learn something. That gave me enough self-confidence to apply to go to graduate school. Without that experience, I would never have gone into science. So it was getting support. And then just to extend this one small bit, when I went to do my PhD, I worked, you mentioned Bob Perlman. Bob Perlman was a spectacular mentor. He was always available to talk. He used to say, I don't care if it's 11 o'clock at night, call me up and let's talk about the experiment. We talked every day. And that also helped my confidence. So I think mentoring means interacting with people and maybe different sorts of ways of doing it, but it can be very helpful. And I was very fortunate that I had people that were very supportive of me. Probably
Thank
too.
you. Fascinating. Fascinating. Let's fast forward to your days in Columbia as a faculty member. How did your Nobel Prize winning work on GFP, how did those insights come about? Yeah, would you like to comment on that?
Sure. Very simply, I was, so as you've mentioned to people, what the Nobel was for was introducing this wonderful protein, green fluorescent protein, as a tool in biology. And the nice thing about this tool is that it's a protein, which means it's encoded by DNA. And you don't have to add anything else. You get the DNA into an organism, it makes this protein. And the protein is fluorescence, which means blue light, if blue light shines on it, it converts the blue light to green. So wherever this thing is, you see green, where elsewhere you don't see anything. And so this serves as a badge, a tag that you can put on anything and watch things happen. When I first heard about this, we were looking at genes that were needed for the development and function of nerve cells. We were finding mutants that were defective in the sense of touch in a tiny worm called Cenorhabditis elegans. And the animals became mutant, that is, unable to sense touch, in two very basic ways. One way is we interfered with the development. So the cells were never made. If the sensing cell isn't made, not going to sense touch. The other way is the cell is made, but the machinery needed to do the sensing would be missing. So we had done a lot of genetic experiments to find mutants that were insensitive to touch, and some were involved in development, and some were needed for sensing touch. And we were starting to clone those genes. And one of the very first questions you want to ask when you're cloning something, a gene, once you know what it makes, you ask, well, genes are not turned on in every single cell. What cell or cells actually uses this gene? And so we wanted to know, for example, was the gene needed, was a particular gene needed in the nerve cell that sensed touch, or maybe in a surrounding cell? We'd like to know where it was working. And one way of getting to find out if where things are working is to be able to have the gene not make it what it usually makes, but have it make something you can see. So wherever the gene is working, it makes what you can see. So we were interested in this problem. I should tell you that there were lots of methods to answer this question. It wasn't that this was a new problem that I had discovered. We knew how to do this, and we were answering those questions. But the other thing about the animal that I'm working on, this small roundworm nematode called Sunrhabditis elegans, is that the animal is transparent. And I went to a seminar and I listened to the speaker, and the speaker talked about green fluorescent protein, GFP. And as I'm listening to the person speaking, I'm saying to myself, I want to know where genes are turned on. And I work on an animal that is transparent. If we were to take that protein and make the gene we're interested in, make that, we wouldn't have to do any fancy preparation or anything. All we'd have to do is shine blue light on the animals, and all the cells that use the gene would light up green. And I just went sort of nuts hearing that seminar. I just got so excited. What a wonderful thing, because I realized that all the methods before had required a lot of work, and they also primarily could only be, the methods required that the preparations go through a lot of work. They had to be fixed. That is, nothing could move around. And then they usually used a chemical reaction that you had to get the chemical into the animal. So you had to basically permeabilize, poke holes in it to get the material into the animal. So you could only do that on a tissue that was already dead. And I realized, wait, if we put this thing in, all it gets turned on by just shining blue light on it. We'll see green. We can look in living animals and watch things happen as they're happening in this. So I got really excited. I do not know what the seminar speaker talked about, because after that introduction, I just started fantasizing about all of the possibilities if this thing actually worked. So I just got really excited about it. And when it was all over, the seminar was all over, and I could finally leave the room, I started calling people and finding out who was in the process of making the DNA that encoded for GFP. And I found that eventually there was a man named Douglas Prasher, and he and I talked together about doing this experiment. Once he got the DNA, we would put the DNA into our animal and we would see how it worked. So I just got really excited in the seminar hearing about it, and then imagining all the things that I could do. I'm not going to go into all the grisly details about why it took us 3 years to finally get back together again. But when we did, I had a wonderful beginning graduate student, Gia Skirkin, and Gia came in and she did the experiment. She put GFP into bacteria, and all of a sudden she had green glowing bacteria. Then we put it in worms, then other people put it in other organisms, and suddenly people started using it like mad, and it was really nice. But the whole idea came from just so one of the questions I've asked myself is, this is not rocket science, right? You work on a transparent animal, you want to know where genes are turned on, and somebody says, look, there's a protein, all you have to do is shine blue light on it and you see green. That connection, why was I the only person in the room that did that, thought about that? And the answer, I think, was that everybody else did not work on a transparent animal. And also, this was not a topic that there were a lot of seminars about. It was not the hottest topic in biology. And so I was just very fortunate to happen to be at the right time, at the right place to hear about this, and then get excited about this idea. And who knows how much other really interesting biology is out there that people are studying that we don't really know about yet? I'm sure there's tons of it out there.
Thank you. You know, Marty, there's a lot of demand, not just nowadays, but always, that the science funded by public money should have value to the public. And what then is the point of working on organisms such as the C. elegans or the fruit fly or the mouse? What value is there in doing it for its own sake, for their own sake? And what value is it there for human health, for example?
So let me start by saying I understand why people would want to see direct application of knowledge. If you have a loved one or if you're yourself are afflicted, you want to get help to them as much as you can. And so people do want to see their lives made better. But the problem in all of this, and I think we've made a pretty good job, done a pretty good job of explaining this to people interested in various human diseases, is we simply don't know enough. And we don't know what the right approaches are of where to look for things. And so every once in a while, I remember hearing about somebody who said this in 2003 in Washington, D.C. There was a big meeting of scientists, and one of the officials got up and said, "Don't we know enough now? So if we just apply everything, we'll be able to cure cancer. So we shouldn't do any basic research anymore." And everybody in the room was appalled because they realized how much they didn't know. You know, in 2000, it was announced 2003, 2003, 2004, there was the completion of the Human Genome Project. So we know what the sequence of all human DNA is. But we still don't know what the vast majority of human genes do. We don't know their biology. There's a vast amount to study. But more than that, we need new approaches. Every time there's a new approach, every time there's some new thought, it brings a new way of addressing a problem. Let me give you an example, not from any work that I've done, but a general example. One horrible disease is sickle cell disease, where a mutation in the globin gene that is used to make hemoglobin, the oxygen carrier in the blood, that molecule, if it has that one defect in it, in all of its what's called beta globin, then the shape of the red blood cells is altered. They become sickle shaped. You know, they look like crescents. And the problem with that is they can't squeeze through the blood vessels. So these people have horrible kidney failure, strokes, other problems that have to do with clogging of blood vessels. It's a horrible disease. So how would you go about trying to address this disease? What would you need? Well, now we have a lot of new methods to try to alter genes. One of the more interesting involves two aspects of basic science. First, there is the general idea of taking what happens in bacteria to protect themselves from viruses, the gene editing mechanisms that viruses come into bacteria and are chopped up. The DNA is chopped up. This has been used as a wonderful tool to change the DNA. It's not easy to change a single defect in the DNA, but people studying what actually happens to blood realized a very interesting thing, that before a child is born, their blood cells don't have beta globin. They have something called gamma globin. And that molecule stops being made after birth, and this new one is made. But the new one is the one with the defect. And then they looked a little bit more. Why is that early one stopped? It stopped because another protein that has nothing to do with hemoglobin, that other protein prevents the first one, the fetal one, from being made. And so one approach that people are doing right now is figuring out a way to prevent that inhibitor. So all of a sudden, in children that after they've been born, they are now making the earlier good hemoglobin so they can live. And it looks like it's actually pretty useful. And there's other diseases. But that's, to me, is a good example of not knowing, not looking just at the end problem, but trying to think, what do we know about the basic development? What other approaches can we take? There have been now 4 different Nobel Prizes that have been given to work using C. elegans. One of those prizes involved the discovery of genes that are needed so that cells die. Cells die as a normal part of development. In fact, it's one of the reasons we don't have a lot of cancer all the time in us, because the cancer cells are sort of made to kill themselves. When that doesn't go around, then they stay around, but that's relatively rare. One of the protections is killing the cell. So understanding how cells die as a normal part of development gives a whole new set of ways of addressing cancer. Can we stimulate those cells to die? Can we turn that gene on so that the gene's on needed to kill the cell so they can just be eliminated? Can we do that? Another one of the prizes went to the remarkable discovery that we know that DNA is 2 strands, the double helix. But a discovery of some friends a few years ago was that double strand of RNA, the thing that's made from the DNA. That double strand is a very potent signal to make sure nothing from that particular gene is ever made. So that's another control point that we did not know about. And there are people that are trying to design drugs to address various human ailments using something that was never discovered in people. The prize this last year was finding that other small RNAs are very potent regulators of development. And this has become a more and more important way of looking at how genes are regulated. And so all of these things allow this. People often talk about mice, yeast, fruit flies, worms as model organisms. I don't like the term. And the reason I don't like the term is because it seems to imply that it is modeling something else, usually human disease. And while that is the case, all of these things I've just mentioned, plus GFP, plus many other things, are all discoveries that were never made or in outside, they were all made outside of human biology. So I think that these organisms should be not called models, they should be called pioneers. They are the ones that are giving us new approaches, new ways of thinking, new tools that allow us to address these problems we all want to solve.
Thank you. That was really very interesting, Marty. I mean, it's also fascinating that the work which allowed the expression of fetal globin in the adult and cure sickle cell disease brings together about 70 years of work from the discovery of fetal globin expression, gene editing, understanding gene regulation. So it has taken a lot of time of multiple labs working on mice, on gene regulation in cells to come up with this. So, lots of integrative things and fortuitous situations come together to make success happen. And that's
there's a wonderful quote from Benjamin Franklin, who in the 1780s was the ambassador to France from the new United States. And he's there watching the ascent of one of the balloons from the Montgolfier brothers, the first hot air balloons going up. And someone in the crowd says to him, "What's the use of that?" And he turns to the person and he says, "Well, tell me, what's the use of a newborn baby? You really don't know where these things are going, but if you don't know them, they're going nowhere."
absolutely. That's fascinating. And, the green fluorescence protein, the ability to label live cells and look at them, has had enormous success, for example, in blood cancers, where you can categorize them into different kinds of cancers, following them, identify them, and then feeds back into sorting cells and treating. So, the complications of application require much integration, and to ask for any specific discovery to have a specific application the next day, science doesn't work that way, nor does application. Thank you very much, Marty. Before we go on to questions, let me ask you a question about science and society. You mentioned before we started that an artist asked you to put litmus paper in your mouth because scientists were neutral and wanted to test that. So how are we to deal with the world around us? Are we supposed to focus on our work? Are we supposed to interact? Are we supposed to constantly give our opinion on matters which we're not expert? What are your views?
So there's a lot of different things there. First of all, we're not the geniuses and everything. We're not knowledgeable about everything. And people often spout off about a lot of things they really shouldn't be spouting off. However, right now, especially here in the United States, but really, I think all over the world, there is a lot of misinformation and disinformation, where either people are misinformed or they're actually actively trying to say things that really are not true. I think it's very important for scientists to explain first what they do, how they do their work, what they are interested in, but also to talk about about various aspects of science, and to make people realize that it's not this esoteric information that no one will understand, but it really is something that is fundamental to our lives. And we should be speaking out. We should be defending it. And I will give an example of what's going on right now in the United States. There have been many statements about vaccines that are inappropriate, that are wrong, where the number of tests of safety and quality control and side effects and everything have been studied for decades. And yet there are people that try to convince others, this is unsafe, you should not do this. Right now in the United States, we have, where a few years ago, we had essentially no cases of measles. We thought we had gotten rid of the disease. It has now come back. and at least one, possibly two children have died of it, it's not something anyone should be proud of. And a lot of the problem is that people, for whatever their reason is, have tried to promulgate that the vaccine was dangerous based on really no information. And I have great fears about what the next pandemic is going to be, what the next childhood disease that we thought we had completely overcome will do to children in the future. I believe scientists should speak out and say, this is not right. This is not appropriate. And we cannot sit on the sidelines. We have to speak out.
Yes, that's a very important point, Marty. Thank you. We are running a bit late, and I must now hand over to Ambika so that she can take questions from the Q&A box and elsewhere. But one quick point, I noticed someone in the box asked whether we could have done this in Hindi. And I think what I will try and do with Ambika is, after this is posted on our website, to have subtitles in multiple languages in due course. Let's try for that. That'll be really very valuable. So over to you, Ambika.
Thanks, Vijay. Yes, and yes, on the subtitles, we'll figure that out. Thanks, Marty. And I think we have a lot of questions. And I'm just going to go with the most simplest question, but very relevant, which is from Snigdha Raj. Sir, you worked on experiments without a set end result when you were younger. Is that something you would recommend to aspiring scientists?
So, one of the things, at least in the United States, that we say to talented youngsters, and it scares them to death, we say to them, follow your passion. Now, if anyone said that to me, and every time I've seen anyone say it to any of the students I've ever seen, the students get a panicked look in their face, and they say, please tell me what my passion is. I don't know, and I don't want to make a mistake. What if I had a passion, but I didn't follow it? My life is ruined. I think that's the wrong thing to say to any student. I think what you should say to students is, be passionate, be excited about what you're doing. You're going to learn something new, and you never know what connections are going to be made. I had no idea I would be interested in green fluorescent protein, but I had been working on an animal that was transparent, and I wanted to know where genes were being activated. The other thing is, it's not that we just work aimlessly, come into the lab and say, I think I'll just do this experiment. We do sort of have a plan. We have an interest that we want to go after. So, for example, when I started working as a postdoc, there really were not, there may have been one gene in one organism, but certainly not in more. People didn't know what the genes were that allowed specific types of cells to be made. And the only sense that we knew the molecule behind the sense was vision. We knew about the rhodopsin in our eyes. We didn't understand how taste worked. We didn't understand how smell worked. We didn't understand how touch, how hearing, any of these senses worked. And the idea that we could find mutants that were defective in touch, that was the project. Could we actually learn about the molecules that allowed an organism to sense touch? And we were able to do that. And so it's not that we just go in and do aimless things. We think about where we want the experiments to go. On the other hand, if you do enough experiments, surprises come up. There's a wonderful quote by the physicist Enrico Fermi that's something, I can't say it exactly, but it goes something like, if you do an experiment and it confirms your hypothesis, you've made a measurement. But if it doesn't confirm your hypothesis, then you've made a discovery. So you do enough experiments and you make those discoveries, and sometimes it leads you into new areas entirely. And one final sentence about this. I'm a geneticist. I look for mutants that are defective in some activity. And then I try to find the gene that's the basis of it. And I will tell you that virtually every time when we find the gene that's needed for sense of touch or the development of a cell, we look at it and go, I don't know what this is. I have to learn about this. So all of a sudden, we're working on a completely different area than we ever imagined we were, because the organism is helping us sort of see where we should be going and thinking about things. So it may seem aimless, but we do, in the back of our minds, we do have a general direction we're going, but we do try to be aware of where the interesting new things might pop up.
Yeah, I think it's a very fine line of going aimlessly and going with passion. So moving to the next question from Ritika Karthikeyan. Sir, you had mentioned regarding misinformation and disinformation is being spread across all citizens. You also mentioned that we scientists must speak out. So how do you think we should convey the reality of these cases?
I think there's many different ways of doing this. Talking to one's parents, one's friends, one's relatives, bringing up the information, talking about what you know, sharing your excitement with the research that you're doing or reading about or thinking about with others. They will get your passion. They will get your enthusiasm. And I think that's important. There are also, we used to have quite a number of these in the United States. I don't know how they are around the world. We call them science cafes, where people, one we had here at Columbia for many years, we went to a restaurant, and the restaurant owner, people didn't go to dinner at 5 o'clock in the afternoon. They went at 7 or 8, but 5 o'clock, his restaurant was empty. So we said, can we use that? We went in, people from the neighborhood would come in. They might have to buy a small dish or something or a drink. And then one person would talk about their science and explain how excited it was, give people an opportunity to hear. But there's also going and talking to government officials. These are our government officials. We should try to go invite them to our labs, offer to come and talk to them about what we are interested in. So I think there's a lot of different outreach opportunities that people can avail themselves of. And especially if people sort of join together, you can get practice on what's the best way of presenting this material or that. And that's also a very useful tool.
Yes, absolutely. And we have a very interesting question, I think, because our audience is so diverse. What does a scientist working in a lab do on a day-to-day basis? All this research papers, experiments, in all of this, how exactly does a scientist earn and make money?
I wish I knew that one. I, so it varies. There are some people that are where what you do is a lot of research at the bench. The experiments are not done in minutes. They are often things that have multiple steps to them, and that you're working on the bench. And gaining the skills to be able to do those manipulations is part of the training that people go through in becoming scientists, is getting used to their hands. In the same way that a physician or a surgeon would have to learn how to do an operation, you learn the various techniques that you can apply to your science. In addition, yes, you do a lot of reading to find out what people are doing. You also do, in my opinion, a lot of talking. I talk to the people in my lab, I talk to seminar speakers, I talk to visitors. I usually learn much more than they learn from me in doing this. And it's very stimulating. I don't know if many people know this. Often people get invited to come and give a seminar, or I'll get invited to go and give a seminar. And one of the really nice aspects of that is not the actual giving of the talk. It's the whole rest of the day, where you go from one person's laboratory to the next person's laboratory all the way through the day, and you find out about the research that they're doing. And it's that interaction and learning about things that I find fascinating. I also teach. I have a course on a second-year course in genetics that I teach. I actually find that very useful, not only because I want to train the next generation of scientists, but also because in simply trying to explain what I do or a paper that I think is important for them to do, I learn more about the paper. I learn a better way of thinking about the material. So teaching is not a one-way thing. It really is an interaction. And so I do that. My typical day is I come in, I usually have too many emails to go through, and but I also try to talk with the people in the lab. And periodically, I will read something and have an idea and get excited about something new and write up about that to myself. Uh, the other thing is maybe a silly story, but I was leaving Columbia, this was several years ago, to go to the airport to go somewhere. And the cab driver picked me up, because I had been picked up right by the university, said to me, "Do you work at the university?" And I said, "Yes, I'm a biologist." And he said, "Oh, you're a scientist." He said, "Now, tell me, do scientists work all the time?" And I said, "Well, before I answer that question, I, I want to know something about you. Is the only thing you do is drive a cab?" He said, "Oh, no, I have a small moving company that I developed. And so when I'm not driving the cab, I'm doing work with my moving company." And I said, "Do you only think about the moving company and its work when you're doing it?" And he said, "Oh, no, I think about it all the time." I said, "You're a scientist. You're thinking about the problems all the time." And lawyers and business people and all sorts of people are, you can't get rid of the problem. It's not that you're working all the time. It's that it's there and it's something you think about. Teaching maybe you do at a very specific time, but you're always thinking about, am I getting it across? Am I trying to what can I do to stimulate the students? There's lots of different things that you do. Um, it's to me sort of a gift that we get a chance to do this, to do something that we get excited about.
I guess it's a way of life rather than, work in every.
But it's not the only thing we do, right?
Yeah.
I have a wonderful daughter who's an actor, and I love going to see her when she performs, and she's 32. And I have interests outside of the lab. We are not hermits. We do lots of things. And I think, if anything, it's because we are excited about science that we're also excited about other aspects of life.
I think that's a very important point that, you see human quality in scientists or humanness in scientists, because
I think
otherwise,
we can't help it,
yeah,
but that's okay.
right. So I'm just moving to a question which is more serious from Arav Gupta. As AI and automation transforms various industries, how do you see science's role in future policy making, particularly regarding ethical AI regulations and scientific integrity?
So this is something everybody is trying to come to grips with. I I know that it's popular to say the term AI. I think it's, it it let me give you this is perhaps a horrible example. I'm going to clean it up a little bit. I I had to have a standard medical procedure done. I get it every 5 or so years. And the doctor, the last time I did it, said, I just want to tell you it's a little different this time. While I'm doing it, we're also using AI. And I almost got up and started cheering, because who wouldn't want the doctor to get a second opinion? The doctor wasn't telling me, we're letting this all happen. The doctor was telling me, I'm doing what I always do, but there's this other thing that might make me look at someplace extra and give a new insight. So I have something that's helping me be more careful. Who wouldn't want that? That's fantastic. Everything that I've read about the use of computer machine learning or other things has always been that it's something that we couple with human ingenuity, human thought, that it's helping us do more things. We don't have a great capacity to keep lots of variables in our heads. If we have a way of being able to have ideas and use something that can help us do this, I think this is absolutely terrific. The problem I see in AI is that when it's only a computer program, there is a danger that people will always believe it's true. And I have an example of that. I have a good friend who has a high school student, one of her sons, and I think he was in high school when this happened. And he wrote an essay for school. And my friend watched him write the essay. She knows he wrote the essay. She gets called into school, and the teacher says to her, I was concerned that some of my students are going to cheat. And for that reason, I took all of their essays and I put them into ChatGPT. And I asked the program, did you write this? And I have to tell you, your son, the program came back and said, yes. Now, the problem with this whole idea is that this program, if you ask it any question at all and ask it multiple times, if the answer is yes or no, half the time it'll say yes, and half the time it'll say no. It has nothing to do with the veracity, the truth of what was going on. It was simply that the program knows, oh, there's a question I have to answer yes or no. Well, 80% of the time, the answer is yes, I'll say yes. Yeah. So but the problem was the teacher believed this. The teacher believed that the student had cheated. And of course, if the teacher hadn't talked to the parent first, and the teacher had gone to the student and said, I caught you cheating. Tell me the truth and don't do it again. And the student would have said, I didn't cheat. And the teacher would have said, not only do you cheat, but you're lying to me. All because the teacher believed the program. I think that's a really important aspect of this. If a teacher is concerned about students cheating on their essays, there's a very easy cure, very easy solution. You have them write the essay in class. You don't have them write it away from, you just have them write it in class. And more than that, I think what would be a terrific idea, I actually did an experiment on this and it worked beautifully. I asked ChatGPT to write an essay. It was wrong in every respect. It was unbelievably incorrect. I think that it would be a great thing to have teachers give students essays that ChatGPT has written and then say, fact check this. Find out what is truthful, find out what's not truthful about this. So I think there's ways of using these. We need to have as much computer assistance to be able to deal with large data sets, with large imaging sets. There is going to be these things. But I think the questioner asked an important thing. What are the ethical questions that should be asked? How should we think about this? And maybe one other thing is, do we always distrust the next generation coming up? I don't think we should, but there, at least here in the United States, whenever they talk about AI, it's always because kids will cheat. I don't think that's really the case. Maybe everybody cheats at some point or another, but I don't think that the next generation is that bad. I think they're actually pretty good.
Right. I think we can have a whole webinar on this aspect. It's a question which many have. The last question, otherwise, we can talk all night, is from Myra Singh. Sir, how did you know research was your calling despite many initial failures? And have your students ever inspired you throughout your years of teaching?
So the answer to the second part is always. They always are the ones that inspire. But one of the most enjoyable aspects I find of having a lab is the interaction with people who are as enthusiastic as I am. That when both people or a group of people realize that they're acting as a team, that they have a common goal to be able to understand some phenomena, or in our case, a particular gene or what it's doing, or this particular type of activity. When you're working with other people, that is very exciting to be part of. So my students often do this, and nobody. The really best situation is when no one feels, oh, my ego is on the line. Who has the idea? Everybody has the idea. You put them commonly together, and the whole thing pushes forward. When did I know I wanted to do research? I mentioned that I worked in a lab in between the 2 semesters that I taught high school. And when I had an experiment that worked, that made me, as I said, gave me a lot more confidence to want to continue. I also, to be very frank about this, I don't like working for other people. I like working for myself. And I feel that this is something that I could do. When I was teaching, it was right after college, and I remember being in a faculty meeting, and some question came up, and I raised my hand, and I said, well, it seems to me logically what we should do is, and I rattled off some idea. And the head of the school looked at me and said, Marty, that is a very logical and very good answer. We're just not going to do it. So we're going to ignore you. And I didn't like that feeling of being ignored. And so being in a lab, I'm still ignored sometimes, but not quite as much. And I did feel that was something I didn't like being told what to do.
Important points to be noted. And I let Vijay wrap it up because I think we're running out of time. We're already 4 minutes over time, though we would like to answer all the questions, and many questions unanswered. I don't know how to get the answers to all those questions. But yes. Vijay.
Thank you very, very much, Marty. And Marty, you were going to say something? Sorry, I interrupted.
No, I was just going to say it's very hard for me to answer all the questions in a succinct way. But if you think there are some that it might be useful for you to collect and email me, and if I can go through them quickly, I'll try to address those so people can get some sort of answer. That would be fine with me.
Oh, that would be wonderful. Thank you very much. And thank you so much for doing this. I know it's been a hectic time for you, and but it's been really wonderful that you could do this. I would also like to thank Ambika and the team for putting this all together. All the people who are at the webinar, thank you for coming. We'll be doing more such webinars, and we all want to hear back from you. And this one will be on YouTube on the webinar site. Hopefully soon we'll start putting annotations and subtitles also. So thank you all very, very much. And thank you again, Marty.
Yeah, and we
Great.
have the next one coming on 2nd May. So we can start. Yeah.
Have you announced the speaker or not yet?
Yes, we will announce it. We'll send it to all the people who have come here anyway. We have email IDs. So
perfect.
Thank you, Marty. Thank you, Vijay.
Thank you, everyone.
Thank you.
Thank you.
Goodbye,
Bye-bye.
everyone.
Bye.
Thank you.
Thank you for the questions. Thank
Bye.
you.